All right. Well, good morning, everyone. Thank you again for attending this morning. If you haven't already, please feel free to say hi in the chat and where you're tuning in from. This is a part of the GBATnet series, which means we like to have a really global presence. We try to set this at a time where we can have as many people attend as possible from all around the world. So please let us know where you're tuning in from. Um, so we know that where we're spreading all of this information to. I am very excited to introduce our speaker this morning. We have Dr. Daniel Sanchez, who's with us today, and he is with the Species from Feces Lab, and he'll be talking to us all about eDNA and how to work with it. So I'm very excited that he's joining us this morning. And at first, just a little bit of housekeeping before I let him take over. So we are still going to be continuing the GBATnet webinar series. So stay tuned for more announcements. Next month, the month of October, the talk is going to be given by Dr. Luis Vasquez, who is going to be a little bit of a different flavor. It is going to be a presentation on the IUCN standard protocols for bat handling. So it's not so much going to be more of this introduction into different fields of research or different types of methodologies, but it is going to cover what the powers that be, not really, but pretty much all of the big minds who work with bat hygiene, working with bats and hygiene and proper PPE protocols. We are going to be doing a talk going over all of that, which maybe doesn't sound the most exciting, but it is one of the most important things that we as researchers and bat biologists need to be aware of. So I highly recommend tuning in for our talk next month. It's going to be, as usual, the first Wednesday of the month, same time, same place. So be looking for those announcements and sign up for the next talk. Without further ado, I would like to pass over the controls to Dr. Sanchez here so he can get started on his talk. So everyone, please welcome Dr. Daniel Sanchez.
Thank you all. I'm just getting the slideshow set up here. Can you all see the screen?
Yes.
Not the presenter view. All right. Wonderful. So yeah, thank you all. It's a real pleasure to be here speaking for GBATnet. My name is Dan Sanchez, and tuning in from beautiful Flagstaff, Arizona, a wonderful mountain town in northern Arizona, kind of close to the Grand Canyon. It is currently 6 a.m. where I'm tuning in, so I definitely made sure to turn off all of my wake-up alarms, which would have gone off in about 15 minutes or so. So right now I'm being fueled with caffeine and the excitement to deliver some information that I hope you find exciting. So what do I do? What do I do in my field? So in my line of work, I specialize in species identifications and detections from molecular sources, namely DNA. And so today, with that in mind, I'm happy to talk about how we can do this kind of work with from samples sourced directly from the environment. And so what I'm talking about is the capture and detection of environmental DNA or eDNA for short. So essentially all this is genetic material in the environment. You can capture it somehow. You can use tools of molecular biology to detect this material and apply it to things like biomonitoring, wildlife occupancy, maybe invasive species surveillance or endangered species surveillance, for example. So when we think about eDNA, we might know, if you've looked into it before, that it's long been famous in aquatic contexts. But it's since gained a bit of traction in terrestrial mammalian settings as well. And now this includes the bat world, much of which is still in its experimental stage, but there's some aspects, such as fecal sampling, that is being pursued more in an applied sense. So agencies and institutions are regularly collecting these samples to identify bats. So just to talk about a little bit where I work at Northern Arizona University as a research associate, excuse me. I work for a team that provides these kinds of species identifications as a research service. And so our team identifies species from a variety of sources, including guano, hence our name, Species from Feces. We also process samples for things like animal diets and develop targeted assays for a variety of focal species. And that's mostly used for eDNA context. So if you're interested in knowing more about what our team does and these kinds of DNA services, I encourage you to check out that QR code there on the left of the screen. I'll show it maybe a couple more times throughout the talk. So that was my shameless plug for our team. A team that I'm really happy and lucky to be a part of. I'm just one small part of this large, vibrant team known as the Bat Ecology and Genetics Lab, led by our very own Dr. Faith Walker. And here in this slide, you can see lab members past and present throughout our team's 10 years of existence. And actually, just a couple months ago, we met our 10-year anniversary as the Bat Ecology and Genetics Lab. When we just started, it was like 4 of us, and then, at various years, we'll have, quite a few people. So my role in the team is I function in our project development, I manage team operations, and I also serve as our team's data analyst. We're also interdisciplinary, so we're a team of molecular ecologists, staff, laboratory technicians, grad students, undergraduate assistants, and wildlife field ecologists, the field division headed by Drs. Carol Chambers and Dr. Jose Martinez. So for this talk, I'm going to talk about kind of 2 components. One half is just going to be an introduction to the field of eDNA, kind of go through the history, some key concepts and considerations. And then we'll start talking about how we might apply this to bats and how it's being experimented on across the field. So eDNA. The one thing you need to know as we get started, you can think about it, is that DNA is everywhere. Genetic materials everywhere. If you take a deep breath right now, you're breathing in the remnants of yourself, your roommates, your pets, and we all experienced a COVID pandemic, so we're all kind of privy about breathing in things that are kind of weird to think about. But the environment's not too different. We can pick up signatures of wildlife from the air, soil, water, permafrost, even other organisms like biting midges, for example, or mosquitoes. We can pick up signatures of wildlife from byproducts we might find, like feces, out in the environment. So eDNA, what is it? Basically these cells that animals leave behind that contain DNA that we're looking for. The DNA could also exist outside of the cell. Maybe it's an organelle like mitochondria that's been expelled. eDNA can also exist outside of a cell in kind of a free-floating manner. And as such, eDNA is, well, exposed to environmental pressures, which can in turn shear and fragment the DNA. So anytime we're detecting eDNA and we're talking about it, we're generally working to detect those small fragments with just enough information to identify the species and in a manner that will kind of give us the most diversity we can get. So for doing eDNA, maybe our primary motive for doing so is to study the ecology of an animal, ecology of wildlife. But it's also worth considering that eDNA has an ecology in and of itself. So there's kind of 4 topics I'm going to go through. When we're thinking about designing an eDNA study or thinking about results we might be reading in a manuscript or a publication, we want to think about, where is it originating from? Maybe out in the water, we're detecting some fish. Is it coming from feces that they're depositing? Is it coming from some reproductive processes at the time, like eggs and gametes? Is it coming from decomposing dead fish, for example? It's going to differ depending on the study system. We also want to think about the state of eDNA. And so this figure is water focused, but eDNA in water, for example, can bind to things like clay particles, and that's going to affect what type of medium you can capture the eDNA on. So you might need a coarse pore filter, you might need a really fine coarse filter. The water that you're working with might be really turbid and really muddy. So you might not be able to get a lot of water through a filter, or it might be really dilute where you could just bring in as much water as you want. So depending on the state of eDNA in the environment, that's going to determine what kind of sampling medium you're going to use to capture it. Then the next two, I think, are ones I think about a lot, and it's in terms of the temporal acuity and spatial acuity of eDNA. So let's look at fate. If we sampled eDNA from some area, how long ago did the animal leave that genetic signature? Was it yesterday? Was it 10,000 years ago? If you're sampling from surface water, fairly recent. If you're sampling from permafrost ice cores, you're going to start picking up ancient DNA. So temporal acuity is important to think about. The other thing is spatial acuity. What I mean by that, for example, if we go out to a lake, sample some water, say we detect gray fox, for example, how certain are we that that's where the gray fox was? Where was the gray fox drinking at that exact same spot? Or was this eDNA coming from a distant tributary and just kind of settling in the lake? So spatial acuity is also an interesting thing to think about in general when we're maybe trying to interpret results or design studies. Just to give you a really brief introduction into the history of eDNA, it really goes back to the 1980s, really starting in the microbial eDNA world. So we're thinking about extracellular DNA from microbial blooms using 16S sequencing. The macrobial world, so eukaryotes, really didn't get their time to shine until the 2000s. And a lot of things happened in the 2000s. For example, we got access to these amazing new DNA sequencers that drastically lowered the cost and raised the scalability of sequencing as much DNA as you want. And so that lends itself very well to community level analyses, maybe to bacteria, maybe for plants if you're getting it from soil. We also got access to things like real-time qPCR sequencers, which I'll talk about in sequencers, real-time qPCR devices or instruments, which I'll talk about later. In terms of the eDNA world conceptually, the first study raised a lot of excitement in the field, where they were able to demonstrate the detection of an invasive American bullfrog, in Europe, straight from water. And so that got people thinking, what else can we get from water? A lot of work was happening between 2008 and 2012, including some of the first sets of eDNA reviews. And shortly after, we can see basically an explosion of eDNA studies throughout the last 10 years. Most of these studies were in like aquatic marine environments, so water. If we think about it, water is a good source of eDNA, particularly in freshwater systems. It offers basically a practical means of monitoring aquatic wildlife, where the conventional techniques were much more difficult and slightly more invasive. It also offered unprecedented sensitivity and ease for things like invasive species surveillance. We can also think of water sampling as an all-purpose approach. So you really only need like a few techniques and you can apply them in similar manners to detect whatever kind of aquatic taxon you're looking for. It's this everything is everywhere approach, which I'm borrowing the words of Dr. Beth Claire though here. If we think about terrestrial vertebrate eDNA, some work was still being done largely in soils and sediments, but the way I see it is that terrestrial eDNA, there were already a lot of really good conventional methods of surveying wildlife that worked a lot better than the eDNA methods of the time. Now things are starting to pick up. There's kind of a new renaissance with terrestrial eDNA. In this sense, we're not necessarily focusing on, everything is everywhere. It's like, where do we look for it? That's kind of the new dimension. So with all these studies emerged a clearer, kind of more honest picture of what was possible. We now even have validation scales that detail what kind of experiments are required for eDNA assays, to function as a de facto survey tool. Many of the promises in these early eDNA reviews happened to be realized, but we also began to note technological and conceptual limitations, and similarly, solutions to these limitations. I won't have time to go into all of these topics, but the takeaway here is that we have a lot of information to establish expectations. And knowing this can help guide our study and survey designs more honestly, and again, aid in the interpretation of eDNA data sets. To talk about the workflow of eDNA, it's relatively simple. You'll see these kind of flow charts across the literature. We start with a question, we define clear goals, we go to the field, we collect some samples, we send them to the lab or take them to the lab for processing. And then we have, a bioinformatician or somebody's good with a computer to identify and pre-process. But after 10 years of doing this kind of work, what I've seen works best is not to view these components as separate entities. Instead, it's important to bring these entities in and integrate them, bring them to the table right at the beginning, set expectations, and refine goals so that they're realistic for all parties. So eDNA is not just something that only geneticists do in a lab. It's ecology, especially when we're focusing on detections. We're working with environments, we're working with habitat, presences and absences, occupancy. The DNA itself is largely just an indicator. So what are these indicators we speak of? What we mean by that is that we're targeting small taxonomically informative gene segments known as DNA barcodes. We don't need an entire genome to identify species. We really only need 80 to 650 bases, depending on what we're looking for. That lends itself really well to eDNA. The types of marker genes you'll select will depend largely on the taxonomic group you're targeting and maybe how small of a fragment we need. So plants, for example, I generally recommend ITS2 and RBCL. We want to pick up plants. For bacteria, the gold standard is 16S rRNA. So if you ever tuned into Melissa Ngala's GBATnet talk, I really recommend checking that out. She goes a lot into the 16S RNA for bacteria work. For bats, cytochrome oxidase 1, abbreviated as CO1, or pronounced in practice as CO1, kind of like co-pilot, and 1, CO1. That's a good marker based on its species level resolution and the sheer amount of barcode references in public databases. There have been initiatives like the Barcode of Life project, which is working to barcode all eukaryotic life on Earth. So CO1 is kind of the gene that has a lot of that information. 16S RNA is also good as a bat identification tool as well. It's really high resolution, maybe just not as many bat references in the system. So as an example of what these types of marker genes is, I have it shown on the screen here. And what you're seeing in the middle are a series of DNA base pairs that we derived from DNA sequencing straight from a fecal pellet like the one shown at the left of the screen. And we can use simply 202 base pairs of this sequence, which is CO1, to identify what bat it came from. So it's kind of forensic. What I have at the bottom is an example of results. There's a number of free tools out there that you can use to identify these sequences. And one of them is the Barcode of Life identification, database identification tool. And what I really like about their results output, especially for demonstration, is just, how certain they are when you do get a species level hit. So I'll read it out to you. The submitted sequence has been matched to Coryneurinus townsendii, or Townsend's big-eared bat. This identification is solid unless there's a very closely allied congeneric species that has not yet been analyzed. Such cases are rare. I love that. There's a lot of other methods as well that you can use toward classification. You can use NCBI's BLAST tool to do local alignments and help guide you through a classification process, either visually, like you're the classifier, or you can feed those results into an algorithm that makes those classifications for you. And quite commonly in our lab, we use a machine learning classifier, which is much more scalable to having lots and lots of sequences we need to classify at the same time. That's 202 base sequence derives from our species from feces CO1 marker. And so this came from an initiative that we published in 2016. And this is kind of where the Bat Ecology and Genetics Lab begun. We wanted to address, a pretty simple challenge. We just wanted, one marker that could identify any bat species in the world. There's like over 1,300. So we went ahead and went for it. There might be situations where you're out misnetting, you catch some bats. They might be hard to identify in the hand. In that sense, you can just take a swab of the mouth, send it to the lab, update your data sheets later. But where it gets its name is that one of the main reasons we wanted to do this is when you go into roosts like caves, abandoned mines, maybe you're checking bat boxes, you might not see any bat. You might see the guano that they're leaving behind. So this is a type of environmental DNA that we'll consider a conspicuous source of eDNA. What we found in that study is that we could actually, with that one marker, 202 bases, resolve 92% of the world's barcoded bat species, which was well over 600 at the time. The other 8% we can get to genus, but in many cases, we can still kind of narrow down what that species is. For example, Myotis auricularis and Myotis septentrionalis basically have the same mini barcode, but they occur in very different locations. One's in the Southwest, one's kind of Midwest, East, and North, and they don't overlap, at least to my knowledge. So if we know where the sample came from, we can make a determination there to the species level. There's some species where they co-occur and you can't necessarily tell apart. There's some species of Myotis, for example, in the US that definitely fulfill that. But we can still at least provide options, of 2 or 3 species that they could possibly be as well. So how do we generate that data? How do we generate those sequences, or at least determine, maybe what bats are an environmental sample, or if specific bat is in some sample? So in eDNA, we typically do so using 2 methods. Both rely on the ability to extract DNA from a sample, whatever the substrate is. If it's a mouth swab, a fecal sample, soil, water filter. And once we do that, we need to make some decisions. Do we want to cast a wide net? Maybe we want to look for multiple taxa, or maybe we're interested in looking for this one taxa. The first step is to generate signal for whatever marker we're looking for. And we do that through the polymerase chain reaction, or PCR for short. Essentially what we do is we have kind of a molecular homing device that targets the region that we want. And we put it through a molecular copy machine. We make so many copies that we generate enough signal to do something with it, to analyze it, whether we want to have enough concentration to load on a sequencer, or just determine if something is there or not. So the first method I want to talk about is metabarcoding. This is a way we can cast a wider net. It's a way we can generate taxonomic mixtures of data. So if there's, for example, on this top panel here, 6 different bat sequences in a sample, we can amplify that, run it through a next generation sequencer, and our data set will look like, one sample, we got 2 or 3 bats with a certain number of copies per taxon. And that's kind of what our data is going to look like. But if we're not necessarily interested in getting, an assemblage of bats, we just want to know if a specific bat's there, we'll actually design our markers to target those specific bats. Again, we'll put it through PCR, but we'll run it through a quantitative PCR machine, real-time quantitative PCR machine. So qPCR for short. Anytime you see this target, we're going to talk about qPCR. Anytime you see kind of a mixture of sequences, we're going to talk about metabarcoding. So as we're amplifying it, we can see it amplify in real time. We add some fluorescence to it. So every time it makes copies, it's going to pass a threshold that can allow us to tell if the bat occurs in a sample or it does not. So metabarcoding and qPCR are some reoccurring methods we're going to be using throughout this talk. So on to the environmental DNA. We've gone through the history and some of the concepts. One of the things I'd like to showcase is our follow-up study to Species from Feces with another aptly named publication, A Fecal Sequel. It was here that we decided to take this at a bigger scale, not just look at proofs of concept. Here, this was a real application where a master's student at the time, Abby Tobin, was going around to all these different abandoned mines across the Southwest. These abandoned mines greatly outnumber natural subterranean habitat for bats, and therefore provide some interesting habitat selection opportunities for a variety of bat species. And so Abby was interested in looking at that. She collected also fecal samples, and we were able to paint a fantastic picture of southwestern bat assemblages. Every bar here is an individual mine. The color corresponds to one of at least 10 bat species we picked up across the landscape. And the size of these subbars here represent the proportion of copies for that particular detection. So you might see a few bar plots here and there. Essentially now, this is regularly used by organizations like Bat Conservation International. We regularly process their samples where we're helping them identify bats that are occurring in various subterranean locations across the US. Another application that fecal sampling is being used for is bridge monitoring. So every month we're processing samples for various state departments of transportation to help them determine if things like endangered species are roosting at these bridges. They might want to know that because there are big efforts right now to repair and maintain our bridge infrastructure. So that's going to help them make management decisions. So in terms of guano, this is becoming more of a standard method as the years go by. But interesting in the study, it started as more of a smaller part, but it really is going to play a big role in eDNA for bats. So this other portion of the study, we were working with Dr. Nancy Simmons from the American Museum of Natural History. She was working at 2 archaeological sites in Belize. We knew these roosts were largely occupied by common vampire bats, but the challenge is they don't leave these cute, adorable little fecal pellets. They leave more of an amorphous blood slurry. And so the goal instead was to sample from sediment to increase our detection success. So soil, sand, and rocks. And we did not necessarily go into this with an eDNA focus, but we became essentially the first or one of the first publications to document eDNA detection of bats from soil, an inconspicuous eDNA source. A few years later, Natasha Sorrell and team did an ambitious study to see if they can detect bats from things like water, soil, and air. So they just kind of went for it. Their approach wasn't metabarcoding. They took a targeted approach. Instead, they developed a qPCR assay, again, presence or absence, for the big brown bat, Feces fuscus. And maybe you're wondering why Aptescus fuscus? It's a very common and widespread bat, across North America, and that's exactly the reason. If you can't detect a really common bat in eDNA, you're probably not going to detect one of the endangered ones that are rare. So what they were able to demonstrate was they could take samples from a moving stream. They could take soil samples outside of a hibernaculum. They even took some air samples from an indoor captive colony at a medical center, and each with known occupation. Each of those samples, they got a detection, which, even though that's kind of a modest set of results, in terms of getting the party started with eDNA, it's actually really encouraging. Since then, there have been a few more studies, and so I'm just going to go ahead and summarize them here. Specifically, I'm talking about inconspicuous sources of bat DNA. So we don't know if bats are there, like if there's no guano or whatever. The first one I want to talk about is water. It's kind of where eDNA is really famous. There's a lot of methods already available for water sampling. For example, some companies offer this eDNA backpack where you can put a tube in the water and just kind of pull it up into a filter. But there are a number of other methods I'm happy to talk about later if you have questions about it. In terms of bat detection, there's a couple studies, one that I just mentioned that went from moving streams, and another preprint that was able to detect bats from water-filled road ruts, out on a landscape. Both of those results were kind of similar. They were still very modest, and right now I'm not exactly sure if there are some downstream applications that resulted from it. So I'm going to rank it, and this is subjective, with one interested Kermit the Frog. The next one I want to talk about is soil and sediment. Simply, you can scoop up some soil, put it in a cup, and there are a number of DNA extraction methods where you can extract DNA directly from that soil material. Aside from the study we did in a fecal sequel and Sorrel study, the reason why I'm ranking it a little bit higher is because Mike Allen and team were able to detect tree roosting bats, at the base of trees by collecting soil underneath it. And they got really impressive results. So I'm going to give it slightly more happy, interested Kermit the Frog here. The next ranking is what we can pull from resources and habitat, where we might think bats are interacting with. Maybe we can sample directly from that source. So along the lines of tree roosting bats, what worked even better in that study was actually sampling directly from the trees. They'd go out with a paint roller, believe it or not, this foam paint roller, start rolling it against the bark, extract the DNA, and they got even higher probability of detecting bats and other arboreal species as well. I'm also going to talk about how we can swab agave flowers to detect nectar-feeding bats in a few moments. And I'm going to give this a more excited Kermit the Frog just because of how impressive those results were. And then finally, I want to talk about this new frontier of eDNA, which is airborne eDNA. And for this, I'm going to give it laser beam eye Kermit the Frog. So in the last several years, there's been somewhat of a renaissance in air sampling as a hopeful analog to the general all-purpose capabilities of aquatic eDNA sampling. Even proofs of concepts are showing that we can potentially monitor vertebrate biodiversity with existing air quality networks. And some of the earliest studies on airborne eDNA, which is really just in the last several years, have been bat focused. How do we collect that? The most effective way, according to the literature so far, is through active sampling. So we can set out these pumps, and that will draw in air, collect on a filter, and we can extract DNA directly from that filter. What hasn't necessarily been published on yet is passive eDNA. So what if we just set something up and allow air to basically naturally settle on some surface, and you set it out for maybe an extended time frame, and we detect bats that way? Stay tuned. We gave it the old college try. First, we'll talk about resources and habitat with an example study that our team did. This was work published in collaboration with Bat Conservation International, namely with Dr. Kristen Lear, who's their agave restoration program director. The project overall involves the detection of 3 nectar-feeding bats from their food sources. And first, I'm going to focus on the endangered one, the Mexican long-nosed bat, Leptonicterus nivalis. It's endangered in the US, listed threatened in Mexico. These listings are in large part due to loss of vegetation. They use as nectar sources, for example, things such as land development and unsustainable cropping practices. Bats are also migratory, so out of 1,300 bat species in the world, fewer than 1% migrate. We don't want to lose this biodiversity. Every year, this species in particular will migrate 1,200 kilometers from central Mexico up into the southwestern US. The thing is, these migratory routes are largely unknown, but theorized. And they're thought to follow the patterns of their food sources along that landscape. As they get more north, agave in particular becomes a more prominent food source. And if you know anything about agave, I really like the tequila aspect of agave. I'm more of a coffee person right now because it's 6 in the morning. But you no doubt probably love these bats for a lot of reasons. So if we wanted to somehow, maybe one day, try and sleuth out these migratory routes, maybe one way of doing that is through eDNA. So for example, swabbing the plants that they're interacting with. If you take a look at this picture, it says 1,000 words. If they're not leaving eDNA behind from these interactions, I don't know what will. So kind of in our first pilot study, we tried a variety of approaches. We actually took cuttings from some of these plants and we tried swabbing these flowers as well. Essentially, the study design is this. We had 2 plants with a known visitation of these bats that we knew from video monitoring. And we'd go out, we'd swab these plants, and the first thing we tried was DNA metabarcoding, which again is resolving taxonomic mixtures. Here we're focusing on mixtures of bats, and we did pretty well. We got almost all the samples, 12 out of 16, detecting the Mexican long-nosed bat straight from the flower. But just because we felt like it, we also added a few other markers. 1 for arthropods, 2 for plants, for example. And just from 2 agave plants, we were able to capture a desert community. We got almost 100 taxa. We got mammals, we got a variety of arthropods, crab spiders, bee species. We even found other plants on this agave flower. This doesn't necessarily mean all of the taxa that we're picking up are directly interacting with agave from natural community processes. However, they could be things like secondary transfer. Maybe a fly is on the poo of the gray fox and it goes to drink the nectar of the flower, for example. Maybe there's dust that's blowing in the air and it's catching inside the flowers. So what I really like about metabarcoding is that even if we did not detect the Mexican long-nosed bat, we'd still have data that might be useful in the future. But if we wanted to make this practical, we'd want to design a targeted assay. And so we designed a targeted qPCR assay for the Mexican long-nosed bat. And we detected the bat in all the same samples, and we did even slightly better. qPCR is nice because it's cheaper, it's faster, and likely more sensitive. We get this eDNA from swabs basically as well as we get it from the flowers, so we can use a more conservation-friendly approach. We also developed assays for the other 2 nectar-feeding bats that visit the United States or migrate to it. And this project was validated recently, actually, with hummingbird feeders. So down in southern Arizona, private landowners, they would put out hummingbird feeders, and these nectar-feeding bats would come and drink from them. There's actually hotels you can go to specifically for that purpose in southern Arizona. Um, but anyway, we developed two more assays. One of the assays was developed by an undergraduate researcher on our team, and Anna Riley. And so our undergrads don't just do, dishwashing or DNA extractions. After 2 years of pretty hardcore apprenticeships, they're doing high-level science. And Anna Riley's not the first to develop one of these assays as well. So we're really proud of that. But how did we do when we tested them against hummingbird feeders? Turns out, whenever a private landowner, sent us swabs, we detect both Leptonicterus yerbabueni and Tyrannicterus mexicana in both of the samples. Arizona, it was a bat party. New Mexico, not so much, but I think this is enough information to say that these qPCR assays can work for nectar feeding bats if we know where to collect the samples from. All right, so that was an example of eDNA from sources of wildlife interaction. So let's go back to this idea of sampling eDNA from the air and all the excitement, Kermit the Frog might have generated in that previous slide. I'd like to direct your attention to Nina Garrett's study entitled Out of Thin Air. I really felt that this was a fantastic project. Essentially, they took miniature air samplers that actively pull in air and deployed them in neotropical bat roosts. Fun fact, some of those parts are 3D printed as well. And so there's some accessibility in getting these kinds of pumps. The eDNA data they got from it really showed a remarkable picture of bat biodiversity in these neotropical bat roosts they were deploying them in. In one instance, they detected a rare bat species in the area that was really only suspected of roosting there. Nina's data confirmed it. So again, they're using active sampling, concentrating airborne particulates onto a filter, and they got great results. Meanwhile, our team, we were asking, could we do the same kind of thing with passive sampling? We just set something out and just detect bats anyway. So we did this work in partnership with Dave Bergman at APHIS. Instead of using a series of impressive pumps, what if we could detect the airborne eDNA of bats with cheesecloth? Not just any cheesecloth, but food-grade cheesecloth. There's a big difference. So good enough for straining food and fermenting veggies at home, good enough for wildlife survey. And by the way, the cheesecloth idea came from a study on koalas for also airborne eDNA related to that study system. And so we also thought, well, while we're there, can we use it to detect bats from open air sites? How we're going to do that? So armed with our field gear and kitchen supplies, Carol Chambers and Jose Martinez embarked on an exciting journey to Nicaragua, where Jose happens to be from. How did we go about doing the open air sampling? First, we probably wouldn't have a ton of luck putting these things out in random parts of the landscape. Instead, we thought maybe we should place them where bats tend to congregate. Anybody here that does a bit of mist netting knows exactly where to find these kinds of sites. So in fact, they chose mist netting locations. This also provided an opportunity to gauge which bats were actually present at the site. Another thing to think about, and what Carol and Jose were privy to, were the contamination hazards with eDNA. For example, airborne eDNA is extremely susceptible to exogenous contamination. And to put it this way, when you get DNA from these samples in the lab, they're not too much more concentrated with DNA than your PCR negative controls. They're just really, really dilute. So in that sense, to start tending to these cheesecloths, only a dedicated technician with dedicated supplies, freshly laundered clothing would do all the cheesecloth work. And they do it about 50 meters away from the actual netting activity. On top of that, we also sought to compare the cheesecloth detection to the leading brand, which is detecting bats from airborne DNA in enclosed kind of neotropical roosts. So how did we do in the open air? Oh, and by the way, we'd set these things up for up to 8 hours. So how did we do? Honestly, not too bad. We got basically 1 or 2 detections of bats from airborne DNA at each site. And if we look at these tables, each panel is a different site with each row a bat species that we captured via misnetting with their abundances on the right-hand column. And eDNA detection on the left, those numbers indicate, how many cheesecloth samples we detected them in. So looking at this for what it is, it really, honestly, isn't too shabby. A lot of the earliest, water eDNA results were still modest as well, and we know how that turned out. But turns out we can just hang up some food grade fabrics out in the open, and you can pick up some bat detections. So I wonder then if we might have better luck with air pumps. Maybe there's a netting site you don't have the logistical capacity to misnet at, and maybe air filters can help recoup some of that information. But again, more work needs to be done, especially on the open air side of things. For the enclosed site, we did considerably better. We actually detected more bat species from the cheesecloth than we did from visual inspections at the time of deployment. So we did a good job there. That might hint that there might be more accessibility into the types of eDNA catchment media that we could deploy in these enclosed roosts. So in closing, I just want to rehash a few things. So we do have multiple sources of eDNA that you can detect bats from. Guano being the conspicuous source. It's already being included in regular surveys. We're processing cave and bridge samples every month. Our team is anyway. The inconspicuous eDNA sources are still experimental, but these early results are continually pushing the boundaries of what we might be able to do. So with that, I'd like to direct your attention to the left-hand side of the screen and shamelessly plug our lab. Follow that QR code, or you can email me if you have any questions that you don't already have now. So thank you all very much for your time.
All right. Thank you so much, Daniel. That was an absolutely fantastic presentation and in such great information on all of those different types of applications. That's definitely a big goal of what we try to offer with these webinars is to actually show applications in ways that different researchers in different fields can actively use this in their own work. So really hit the nail on the head with that.
Awesome, thanks.
Would love to open up the floor. If we have any questions for Dr. Sanchez, please put them in the chat or feel free to unmute yourself.
Hi, everyone. Good morning.
Good morning.
Hello, good morning.
Let me open my camera. Hi, my name is Alejandra Camacho from Ecuador. I work at the Museum of Zoology here at the Battle University. As I understood, you offer your services at your lab, right? So,
Yes.
have you worked with institutions from the global south, from South America, maybe? So kind of because I'm interested, but always in South America is difficult to get funds and access to
Right.
this kind of services in the US or in Europe.
Yeah, I totally understand. Yeah, so we mostly work with a variety of organizations, institutions, and agencies throughout the US, but we also work with folk from Canada, and we've even worked from groups from Rwanda, for example, so on the African continent. We're actually doing some work with some researchers from Guatemala right now, and it's more of a population genetics approach, more experimental. We're trying to develop. So yeah, we're pretty flexible, and we'd be happy to give kind of a consultation on, kind of the nitty gritty of how, the permits go and sending the samples and stuff like that. It's best to just go ahead and contact us, and we can offer some more detailed insight there. But yes, we do work with the international community, but largely focused in the US, simply because, it's proximity.
Yes, but thank you so much. Excellent presentation. Thanks so much for that.
Thank you very much.
Yeah, and I can kind of commenting on that same point, I can attest. I worked with the species from Feces Lab for my own PhD work, and I'm located in Canada. So working with them, they made it really easy and accessible and willing to kind of like work with whatever accommodations need to be made, which in my case, working from Canada, there's a little less pushback when it comes to like border issues and mailing things. So it's always going to depend country to country. But speaking like from a personal context, I worked with them from my own lab in a different country, and it worked out very, very well. I'm very grateful to have worked with the lab.
Nice. Thanks, Dana.
Another shameless plug for you. There you go. I'm
Yes,
biased.
yes, never have enough.
All right. Well, if we don't have any more questions, if we do, like, please chime in now. But otherwise, we can wrap up for today. Yep. Another comment. Yeah. Oh, greetings from Paris. Hello.
Hi, thanks, Antonia.
All right. Well, thank you everyone so much for coming in today. We had such a wonderful representation from all over the world. So thank you everyone for showing up this morning, this afternoon, or this evening. And look forward to hopefully seeing more of you at future GBATnet webinars. So thank you everyone for coming today, and stay tuned for more.